Okay, so this is going to cover chapter 15, sections 3, 4, and 5. So the first topic is on linked genes. Oh, where's my recorder? Already having problems. Oh, I did not want to do that. Already having technical difficulties. Chapters 15, sections 3, 4, and 5. This new topic on linked genes is going to be a little bit more challenging because it's not something we've ever really thought about before. Genes located near each other on the same chromosome can be inherited together in a genetic cross. These are linked genes. Now, don't confuse this with sex-linked traits. Sex-linked traits are traits that are carried on the X and Y chromosome. Linked genes occur on any of the autosomes, which are the chromosomes are not X and Y or XX, not the sex-linked or sex chromosomes, and they're inherited together because they're so close together on the same chromosome. Mendele's original law of independent assortment assumed that genes were located on different chromosomes. But we know that there are some genes that are located on the same chromosome, and if they're really close together, the odds of them being inherited together is pretty good. You can read about Morgan's experiments with fruit flies, where he discovered linked genes with eye color and wing size, and that these two are usually inherited together. And I have a diagram of this. You can see here that these two traits are pretty close together on the same chromosome. And so when they duplicate, now I don't want you to be confused by the way they write the traits here. This big B plus or little b plus, that is a dominant trait. It's a wild type. And so this organism is like this, heterozygous, and then heterozygous for the wing. And so this is kind of like writing big B, little b, big B, little b. Not really sure why they don't fly, they just don't. It has to do with the wild type, the dominant, and the pluses and minuses. And I don't really know exactly why that's like it, but anyway. So DNA's chromosomes have replicated. So you've got two copies. They're the same here and there. Okay. And then this one copied itself too. So now you've got actually four copies of each of these alleles. Meiosis 1 happens, and notice this crossing over right here. So now the big B and the vestigial wings here, but the big B and the, well, this is the dominant wild type normal wing, sorry. The gray body and the normal wings, but now we've got the gray body and the vestigial wings together. And you can see the crossing over that results here. So this is okay. Recombination happens, and you've got all of these options. You would expect then that when crossing over does happen or things are inherited on separate chromosomes, that you're going to have about an even distribution in the offspring, should be about 50-50. But that doesn't happen in genes that are so close together that they're usually inherited together. A lot of times, you actually see higher than normal number that look like the parents, that have the parental phenotype. If you go back, we had gray normal and black vestigial, gray normal and black vestigial. And then the recombinants are the ones where you see that crossing over happening. And there's less of these. And so this ratio is a little different than what you would expect if you were thinking in terms of Mendel. And you can actually go through if you want and take these two sets of parents, and you can predict what for Mendelian genetics, what should have happened, okay? And you're not going to get this ratio right here. This is a little off, okay? We can actually calculate how much recombination happened by adding together the recombinants here, and then dividing that by the total number of offspring. So you add all of these, multiplying by 100, and in this case, it's 17%. The lower the recombination, okay, the more likely that these genes are inherited together. So the rule of thumb is if recombination is less than 50%, that means that they're linked. Okay. That means this number is going to be greater than 50%. And then the recombination is going to be less than 50%. That's how we really definitively determine if those genes are inherited together or not. What are recombinants? Are the offspring that express new combinations of traits that were not expressed in the parents. And we expect that with Mendelian genetics. Recombination, well, how do we get that? Crossing over can account for recombination of linked genes, which you don't have as much of that, and so that's why the number is low. Okay, the recombination amount is low, because crossing over is going to only be the only accounting for why they don't look exactly like the parents. It's not going to be that they independently assorted, because that doesn't apply in linked genes, because they can't independently assort. They are stuck together. They're so close together on the same chromosome. From there, scientists can actually construct a linkage map based on how far, and we call those map units, genes are from each other on a chromosome. Keep in mind this, map units are not real units. Okay, it's not like saying, I am 1 mile from you, or your house is 1 mile from my house. It is not a real number. It is an arbitrary number. It is a unit of measure. So we can't actually determine how physically far they are, but we can know something like if 2 genes are 9 map units and another set of genes are 10 map units, we know that these are closer together than these. Okay? That's what it tells us, is how far apart they are. They're arbitrary. They're not real numbers. But it gives us the ability to predict the order of genes relative to one another on a chromosome. The further apart the genes are, the more likely their allele combinations will be recombined during crossing over. And if you think about that, if they're pretty far apart, then crossing over would be more likely to happen. And this just shows in a sperm cell, for example, and you can read all this side stuff, but it's like C and C, they're on separate chromosomes, so they should assort independently. They're not necessarily always going to go together in the offspring. However, these 3 are right here all together, so they're pretty much always going to go together because they're so close together. And they're on that same chromosome, and they're probably not going to do a lot of swapping during crossing over because they're so close together. The closer they are together, the less swapping over that will happen during that. So recombination frequency will be 50% when 2 genes are located on different chromosomes, because they're the law of independent assortment. When 2 genes are close together on the same chromosome, they do not assort independently and are said to be linked, whereas genes located on different chromosomes assort independently and have a recombination frequency of 50%. Linked genes have a recombination that is less. That's how we determine whether they're linked or not, is we look at that recombination frequency. And if it's less than 50, we say they're linked. And this gives you another example here of recombination and how you can get some crossing over that can account for your recombinants. But when your parental numbers are greater than the recombinants, and remember, recombinants are just anything that doesn't look like the parents physically, then if that recombination amount is less than 50, we would say they're linked genes. So when you get weird numbers like this that would not follow a normal Mendelian pattern. And then this is a linkage map. So from here, we can determine based on the map units, okay, or the recombination frequency, we can turn those into map units. So we would say 9, 9.5, and 17 map units. We can actually put these in order, and you're going to have some practice putting these in order. And that doesn't mean necessarily that these are a certain distance from each other. It just means that on a chromosome, this one, and this one are in that order. This one is in between the others. Okay, and then the last thing that is really not that difficult is alteration of chromosome number and structure. Non-disjunction occurs, accounts for changes in chromosome number. This is where either homologous chromosomes or sister chromosomes do not separate properly in meiosis, and I should show this. So in this, here, you had non-disjunction. Okay, these guys accidentally did not separate like these two did. They went together. And so now we have a cell with extra chromosomes and a cell with not enough chromosomes. And so normal number, but extra. You're not supposed to have those. And then these are lacking strands. And then this is showing non-disjunction over here, where it doesn't happen in meiosis 1, it happens in meiosis 2. So these go together and this. And so you have an abnormal number here. These have an extra, and this has not enough. Okay, so it can happen in meiosis 1 or 2. So that Aneuploidy, monosomy, and trisomy. This is where like there's one or two or three extra chromosomes. Not a lot here. Most of the time in humans they don't develop. This is where they think maybe a lot of miscarriages happen is abnormal chromosome number, but they don't really know because usually it's not even detected. The egg never develops, never triggers a pregnancy or anything like that, and so we really have no way of knowing how many zygotes or fertilizations happen with this type of thing happening. Trisomy, the best known one, the only one known to survive in humans, would be Edwards syndrome, which is trisomy 18, and Down syndrome, which is a trisomy 21. And these Edwards syndrome are babies dying, I don't know, pretty young. They don't usually survive the first year, I don't think. Down syndrome is the only one that can survive for quite some time. Polyploidy is one or more complete sets of chromosomes, and so instead of having 2 in, you might have like 4 in, or 8 in. That's really only going to happen in plants, in which new plant species can actually come about through polyploidy. And then there's alterations in structure, not necessarily number. You can have a deletion of a partial chromosome, duplicating part of a chromosome, inverting, or translocating. So let's look at some of those. And you're going to have to read about this. I think that's part of your chapter reading. Oh, here's Down syndrome, trisomy 21. Everybody, there's 2 copies, one from mom, one from dad of every chromosome, sorry. And what creates this characteristic Down syndrome appearance and the mental retardation is whatever genes are on that chromosome 21, when you add more of them, it's going to create those characteristics. So let's look at the alterations in the chromosome. So a deletion here, say this section got deleted, whoop, well, it's not here anymore, so whatever genes were there are no longer going to be in the offspring. The duplication means you got this and you duplicate it, so now you've got more genes than you should have. Duplicating an entire chromosome here is a lot of extra genetic information. So again, only 21 and then a trisomy 18, I think, are known survivors in humans. An inversion, so here's your gene order, and whoop, it was inverted, so now it's BCD, now it's DCB. And a translocation, so here it is, and here it is, and oh, we swapped those up, and so now they're on different chromosomes. Genomic imprinting. Variations in phenotype depend on whether an allele was inherited from the male or female parent. These genes are on the autosomes, occurs during gamete formation, and results from the silencing of a particular allele for a certain gene. Relatively new and only a few genes have been identified as being inherited this way. And the book explains an example of mice. And again, I don't know much about this because, again, it's something fairly new. It's been a long time since I've been in college. Inheritance of organelle genes that are located, we call it chloroplasts, and they're trying to have their own DNA. They're not subject to Mendelian laws because they don't follow the same patterns of inheritance. They don't separate independently because they're not made in meiosis. They're actually copies sent through the mitochondria and the chloroplast. Products of most mitochondrial genes make up the protein complexes of the electron transport and ATP synthase. A few disorders are actually linked to these, and remember the mitochondrial DNA is inherited from the mother directly, and that's interesting, or makes sense, I guess, that they have to do ATP synthase and the electron transport chain because that's where cellular respiration happens. And that's the end of that.